Hello, good evening, Christina. Good evening, Dr. Anil Kumar, and good evening, Sabu.
Good evening.
Yeah.
Good evening.
Good evening to all.
Can we start? Can we start?
Yeah.
Okay. Actually, I'm sorry, my video having some issue. So it's no issue. I can go without video. So let us start. Yeah. Good evening to all panelists, speakers, and audience. Of course, it's good evening and good morning, good afternoon. Let me introduce, this is the continuation of our session, Southern Ocean Plankton Diversity, Food Web Ecosystem and Biogeochemical Cycles. So it is the last part of our session. And morning we had a very good session of this Plankton Diversity, Food Web Ecosystem and Biogeochemical Cycles. We have at present 6 oral presentations followed by 6 posters. So for the posters, if anybody want to present their poster, then we can allow 2 to 3 minutes for the poster presentation. And all the speakers must be aware that about our presentation. Each presentation is 15 minutes. Out of that, 12 minutes is presentation followed by 3 minutes question and answers and discussions. So myself is from NCPOR, RK Mishra, and Anil Kumar is from NCPOR, and P. Sabu is from NCPOR, and then Christina Weldrick from University of Tasmania.
Hi, everyone.
So please, this is some information. This is information about the all the registered participants. The questions can be asked in the question answer box function in a Zoom function. And then session will be recorded to be made available to registered participants in the past event section of the events platform. The organizers are also planning to upload the recording in SCAR YouTube channel and websites. If anyone doesn't want their presentations to be made available, then they can intimate to the SCAR 2022 @albonmeetings.com on or before 12th August to opt out. So the first speaker, I can invite to the first speaker is a Nicola Halsey, and the title is Antarctica Krill Efforts Here Superbug, Behavioral and Swimming Kinematic Responses to Chemical Fatigue and Physical Stimuli. So welcome to Nicola. Please start your presentations. Nicola is there? So the first presenter, Nicola Halsey, I think we are not seeing to
Yeah, I could not find her actually.
IT team, any information from their side?
Nikolai has not yet joined.
Pardon?
Mr. Nikolai has not yet joined.
Not yet joined.
Okay, so then can we go to the second speaker? So
Yeah.
at last again we can invite to him.
Yeah.
So the second speaker, Martina Massoni, and her title is Phytoplankton Variability in near-shore regions of the Western Antarctica Peninsula in relation to a temperature gradient.
Hi, I'm here. I just can
please.
turn my
Please
video
come and
on.
start presentation.
It doesn't let me start my video on. I don't know why.
Is it your video? Okay.
Listen. Can you see my screen?
Yeah, it's visible.
Okay, I'm gonna do it without video. I don't know what it's now. Say you're gonna start your video because the host has to stop it. Hello?
Hello, it's visible. You can start here.
Okay, so
You
I
can
don't
go
turn
for full
on my,
screen. Yeah, you can go for full screen.
okay. So hello everyone, my name is Martina Massioni. I'm a PhD student for the University of La Plata in Argentina, and I'm gonna talk today about phytoplankton variability in the nearshore regions of the Western Antarctic Peninsula in relation to a temperature gradient. So we're gonna focus on the Gerlach Strait. The Gerlach Strait is a narrow channel that separates the western coast of the Antarctic Peninsula from this archipelago that is called Palmer Archipelago. That's made of these two big islands, Ambers and Brabant, and some smaller ones. And so the Gerlach Strait current goes from the south, entering through the Bismarck Strait and going up to the Brainfield Strait that's up here, and some waters that also enter to the Scholar Channel from the Bellinghausen Sea. So the characteristic of the area actually favor the development of different phytoplankton blooms during spring and summer months, which are enhanced by the reducing mixing process characteristics of protected coastal areas. So it's a very productive coastal area. And some researchers have found that there is a thermal front in the surface waters of the Gerlach Strait, more or less here in 64.5 degrees south, that has been recorded many different years and many different summer months, December, January, and February. And in 2018, Mendes et al. Found that this Gerlach front not only separated warmer waters to the north and colder waters to the south, it also separated two different assemblages, north and south. But the actual effect of the thermal front on the phytoplankton composition has not been assessed for the entire growing season. So we are interested in the nearshore regions of the Gerlach Strait. We are going to focus for this study in 6 nearshore regions: Sierra Cove, Wilhelmina Bay, Cooperville Island, Danko Island, Nico Harbor, and Paradise Bay. So the studies in the nearshore regions are really scarce, and there's no well understand how the Gerlach Strait waters and the waters from the nearshore regions interchange and work. So if we place the front that hypothetically is around here, we can divide these areas in northern areas and southern areas. So here we assess phytoplankton composition and succession through the spring summer months, November to March, in these 6 areas to try to understand the influence of the surface waters of the Gerlach Strait on the phytoplankton communities in these nearshore areas. So the study was carried out in this citizen science project called Fjord Phyto. If you want to know more about the project, please visit our website. And this project has been running since 2016. And for this particular study, we are only going to focus on the 31st seasons, 2016, 2017, 2017, 2018, 2018, and 19. So we analyzed surface water samples under the microscope. We also analyzed sea surface temperature MODIS Aqua level 3 images. And in order to assess the differences between northern and southern areas, we performed a non-metric multidimensional scalar analysis, or MDS, that was performed in our studio. So we analyzed the images from MODIS Aqua, and then we could evaluate 10 years of what happened with the temperature in the surface waters during in this area. So we were able to identify the surface gradient and temperature gradient, where warmer waters are in the north of the Gerlach Strait and colder waters are towards the south of the Gerlach Strait. But the actual location, like the line that divides the warmer and the colder waters, was difficult for us to identify with this kind of images. We also evaluated what happened during the 3 particular years that we sampled. We could see that 2016, 2017 was actually a very warmer year. And 2017, 2018 was a little bit colder, and 2018, 2019 was the coldest of the 3 of the years. So it's like cooling pattern going on. But in the 3 years, we were able to identify this gradient during summer months between December and February. So what about the phytoplankton composition? That is what we were interested about. So we pulled together all the samples from the month from December to February, the 3 years, and we performed this MDS analysis, and we observed that the samples grouped in 2 different groups. This group A that is in green here is pulled together all the samples from the 2 first years from the southern areas, and it was characterized by big diatoms like centrix, Odontella floshi, Costina discus, Eucampia antarctica, and also these Pyramimonas that is a parsinophyte. And the group B have pulled all the samples that were taken in the northern areas during the first 2 years and all the samples from the 3rd year. And it was characterized by the presence of small flagellates, small Fragiliopsis, small centrix, cryptophytes, and some corythrum. So what happened when we see the whole picture, what happens between November through March, the 3 years? So we have here the relative phytoplankton biomass of this year. And the composition we can observe during November is quite similar during the 2 areas. And well, this makes sense because we didn't observe this gradient in this month. During December, the composition is actually quite different. We can see in the northern areas, cryptophytes, this brownish color, are the ones that dominate. And in the southern areas, the green color, that are the diatoms. So all the greens are diatoms, different greens are different taxa. The diatoms are the ones that dominate in the south. During January, the cryptophytes decrease in the northern areas, and some diatoms took over, but not the same diatoms that we observed in December in the southern areas. And what happened in the southern areas is that parsinophyte, these big Pyramimonas, took over, and they are really, really important. And during February, in the northern areas, we have more dinoflagellates and diatoms. And in the south, some parsinophytes still stay, and some diatoms are also present. That happened the first 2 years. But what happened with this strange 3rd year that pulled together with the samples from the north area? So we can see here that this year was actually quite similar between the northern and the southern areas, because the cryptophytes were the ones that dominated. We couldn't observe in the southern areas the parsinophytes, Pyramimonas, that were actually important, and the small flagellates are also another important group. This was also a year that had a very low biomass. So what we hypothesize is that we have 2 different phytoplankton succession, north and south of the Gerlach Strait. In the northern areas, so in both areas, we observed this basic assemblage that was made of small flagellate, cryptophytes, dinoflagellates, small parsinophytes, and a small Fragiliopsis. And this is like the persistent assemblage in the area where other groups are included. So we observed a similar composition during November, low biomass, and this corythrum dominating. Then on December, we can see the big difference. Here we have cryptophytes and a small flagellates, And in the southern areas, we observed these large diatoms. During January, some diatoms took over in the northern areas, these small centrics and ketoceros, and in the southern areas, we observed the pyramids. During February, the biomass decreased a lot, and it was more or less the same composition. And during March, even though we only observed that in the southern areas, we observed like this basic assemblage that is common from the autumn and winter composition in the area. So during the summer months, we were able to identify the surface thermal gradient established north to south with warmer waters to the south and colder waters to the south. And we believe that this gradient is the reflection of this wide frontal area that agrees with previous records of the literature that have found this front located around 64.5 degrees south. The frontal areas have many implications in the biology. They are known as the biodiversity hotspots, and often the fronts are better defined by changes in the composition of the biological communities, like we found here. And also we found that Giraldo et al. Found two different copepods assemblages north and south in the Gerlach Strait, meaning this frontal area might have several implications for the rest of the food web, not only the phytoplankton. So this is the first study to assess the phytoplankton composition through the entire growing season in the nearshore areas of the Western Antarctic Peninsula between 64 and 65 degrees south. So here we present a complete phytoplankton succession during spring and summer, and our results provide evidence that phytoplankton composition and carbon biomass between the nearshore waters of the Western Antarctic Peninsula are closely related with mesoscale features created by physical oceanographic characteristics in the Gerlach Strait, like this thermal gradient. Thank you very much for listening, and please contact me if you have any question, and be free to follow the project and send emails.
Thank you, Martina, for your excellent presentations on phytoplankton at the Western Antarctica Peninsula in relation to temperature gradient. So I'm not seeing any question in question box.
Shall I ask one question?
Yeah, please.
Okay. Very nice presentation. And yeah, I would like to ask you about, you are telling about the temperature gradient between north and south. What is the temperature difference actually you are getting there? And second thing is that you are telling about a thermal front there. A thermal front, is it a southern boundary? Because we are observing so many fronts when we are moving towards south from north. That is subtropical front, subantarctic front, polar front, then southern Antarctic circumpolar current front, and southern boundary. Whether this thermal front is related to that or a different front. That is my question.
Oh, sorry, I didn't catch the whole question. Maybe the first one. You're asking about, and the second question you're asking, what are the difference between the polar front and this particular front?
No, this front is the southern boundary, because the maximum when we are going to south, southern boundary is the last front what we are observing. Whether this front is related to southern boundary or a separate front, this one.
Well, this front, I didn't define this front. I found it in the literature that other authors define this front, but a difference between the northern and the southern areas are about 1 degree Celsius, and it is made of a air-sea interaction that made that the layer heat up, and that's why also it only formates during the summer. It's not something that is persistent during the whole season. I don't know if I answered your question.
Okay, thank you. And this is the temperature gradient is 1 degree, you are telling, right?
Yeah, the difference between the north and the south are up to 1 degree Celsius.
Any other areas this front has been reported?
Well, if I think there is only a very located feature that only happens during summer month in this particular restricted area. Like it doesn't relate what happens on the Bellinghausen Sea that is open area, for example.
Okay, okay, thank you, thank you.
No, thank you.
Yeah, so there is no more question. Anybody wants any question? Okay, so then we will go to the next speaker. The next speaker is Lucy Borrow, and the title is Chlorophyll A within East Antarctica coastal pollinators during the austral, autumn, and winter, collected from animal biotelemetry. Lucy.
Yes, hello.
Are you ready? Okay.
Yes. Can you see?
Yeah, is visible.
Okay.
Is visible.
Hello everyone, I'm Lucy Bourreau, and today I will present you the results I obtained during my internship that I did for my master's degree. This presentation is entitled Chlorophyll A within East Antarctic coastal pollinators during the austral, autumn, and winter, collected from animal biotelemetry. So East Antarctic coastal pollinators are defined as persistent and recurrent areas of open water that are located between the coast and the sea ice. Here on the left, you have a map of the East Antarctica with in blue the contours of the pollinators we used for this study. In spring, pollinators are the first polar marine systems to be exposed to solar radiation, so that creates the emergence of a phytoplankton bloom. And it's also the season when the surrounding sea ice start to melt. During summer, the phytoplankton bloom is increasing, and this is attracting many higher trophic levels like whales and seals. And the surrounding sea ice is still melting. During fall, the phytoplankton blooms start to decrease and the production of sea ice restart with a release of salt and a loss of heat. But during winter, all pollinators are completely inaccessible by boat, so this is considerably limiting our data. We know that we still have the production of sea ice, but the data about the phytoplankton blooms don't exist from in situ observation. So how can we collect in situ data within pollinators during the months of fall and winter? The answer is to use biotelemetry. So on this picture, you can see 1,000 elephant seals that is equipped with a sensor. So we use 1,000 elephant seals because they are sentinel species, so their presence will represent the presence of lower trophic levels. The sensors are catching data about temperature and salinity of the water, and those data are then transmitted via the Argos transmission. And since 2011, some 1,000 elephant seals are also equipped with fluorometers, so they catch the fluorescence of the water, and this is a proxy for the chlorophyll A concentration. So the objective of this work was to analyze for the first time the primary production within pollinators with the first in situ data that were recorded via biotelemetry during fall and winter. During this presentation, I will start to describe the seasonal signal of chlorophyll A, to then identify the drivers of the signal, and then I will present you a way to predict chlorophyll A profiles from temperature and salinity profiles only. The data I used for this project were mostly composed of in situ profiles of fluorescence, salinity, and temperature that were recorded by 9 individuals that visited the Cape Darnley, Barrier Bay, and Shackleton pollinia. This representing 2,060 profiles recorded between February and August. And today for this presentation, we will focus on the results we obtained for the Shackleton pollinia. So first, to describe the seasonal signal of chlorophyll A, I want just to introduce the hypothesis we had. So this is a simplified representation of the water column during spring and summer. We have a strong stratification, and at the surface, the mixed layer with the chlorophyll A, the phytoplankton bloom. And during fall and winter, with the increase of wind, we have a deepening of the mixed layer. So we hypothesize that we will see a decreasing and a deepening of the signal following the mixed layer. So these are our results for the Shackleton polynya. Here on the y-axis, you have the depth, and on the x-axis, the chlorophyll A concentration. Its color is representing a month, and solid lines are the mean profile for each month. And the horizontal line represents the mixed layer depth. So what we can see here is that we still have a significant signal of chlorophyll A in April, so during fall within the polynya. So for the first time, we are able to say with in situ data that we still have a signal in the polynya during fall. But what we can also see is that we have the peak of chlorophyll A that is located below the mixed layer depth, and this is in contradiction with our first hypothesis. So we want to understand why we are observing that. On this map, you can see the polynya with all the profiles that were recorded here. And the profiles are colored according to the day of the year. And this is the eye chef of the Shackleton polynya. And what we can see is that all the profiles that were recorded during fall and winter were recorded along the eye chef. So that means that the southern elephant seals seems to be attracted by the eye chef during fall and winter. And we also want to understand why. So to summarize the results, we have a maintenance of the signal through the fall season, but also a signal that were below the mixed layer in April, and a potential effect of the eye chef. So now we will identify the drivers of the signal. So we have all the profiles that we recorded in this polynya, the profiles of chlorophyll A, salinity, and temperature on 161 depths. So those are our variables. And on this matrix, we can apply a principal common component analysis that will give us two main results, the principal components and the vertical modes. The vertical modes aim to visualize the effect of the principal components on the mean profiles of chlorophyll A, salinity, and temperature. So we applied the PCA on the matrix, and for the first principal component that represents 41% of the variance of the matrix, we observed that this principal component was reflecting the season. So that wasn't surprising, since that through time we have a release of salt, so an increase of salinity, but a decrease of the temperature and a decrease of the chlorophyll A signal. So what we want to understand now is what the second principal component is representing. This PC2 represents more than 28% of the variance of the matrix. And here is the same map as previously, but this time all the profiles are colored according to their position along the axis. So in red, you have all the profiles on the positive side of the axis, and in blue, the profiles on the negative side. What we can see is that the axis is discriminating a specific area here along the ice shelf. And this area corresponds to the profile that we're recording during April. And in April, we had a chlorophyll A peak below the mixed layer depth that we want to understand. So to understand those observations, we look at the vertical mode. So we still have the depth on the white axis and the chlorophyll A concentration, salinity, and temperature. The black line represents the mean profiles of each variable. The red one, the positive effect of the axis on the mean profile, and in blue, the negative effect of the axis on the mean profile. What we can see is that salinity represents more than 85% of the variance of the axis. So that means that salinity has a strong impact in this polynya. And our specific area is characterized by lower salinity than the other parts of the polynya. It's also characterized by higher temperature at depth and higher chlorophyll A concentration. So we have two main interpretations to explain that. Those interpretations are based on the literature, hypothesis, and suggestions. So we have one dynamic and one biological. If we have warm water intrusion at depth, so we can have a melting of the ice shelf that will release fresh water, so decrease the salinity, and explain why we can find low salinity in this specific area. But the melting of the ice shelf can also release iron, and those two together can create an optimum between nutrients at depth and light at the surface, and explain why we can have an increase of primary production below the mixed layer depth. But we can also have a strong stratification of the water column during spring and summer, and so have a depletion of the surface nutrients that will arrive sooner during the season. And so large diatoms that can control their buoyancy can be attracted more at depth, and explain why we can see a peak of chlorophyll A below the mixed layer depth. And of course, those two interpretations are not exclusive. So until now, I'll present you the results we had for the Shackleton Polynya, but each Polynya is unique. Some have an ice shelf like this one, but some don't. Each Polynya have a typical topography, all of that. So if we want to have an ecosystem index of Antarctic Polynyas, for example, we really need to increase our initial data set to be able, for example, to create maps of primary production in all East Antarctic Polynyas. So to remember, we had 9 individuals that recorded 2,060 profiles of chlorophyll A temperature and salinity. But in total, since 2004, we had 142 individuals that visited 19 Polynyas, and this is representing nearly 47,000 profiles. But those individuals were not equipped with fluorometers, so they only catch temperature and salinity data. So we create a neural network to understand the relation between chlorophyll A temperature and salinity to be able to predict chlorophyll A profiles on those other temperature and salinity profiles. So this is the main results of our neural network. On the y-axis, you still have the depth, and on the x-axis, all the profiles together. On the first panel, the observations. On the second, the prediction by the neural network, and then the difference between observation and prediction. So what we can see that the neural network tend to underestimate the signal. We have mostly a blue difference, but the shape of the profiles is well represented, so we can identify where the peak of fluorescence is. So this is a really good prediction. So to conclude on this work, for the first time with in situ data, we were able to say that there is maintenance of the chlorophyll A signal through the full season. And this is suggesting that Polynyas are a biologically attractive system. We also identified unsuspected drivers of this signal, like the melting of the ice shelf. And we were able to make reliable prediction of chlorophyll A profiles from only temperature and salinity profiles. And this offered the opportunity for larger scale studies. Thank you for your attention, and I will be happy to answer your questions.
Thank you, Lucy. It's a really very nice presentation and very informative regarding the prediction of chlorophyll A in coastal Polynyas. So you have taken a large profiles and all, that is also very good. Just wanted to know what is your percentage of underestimating of this prediction then observations?
Sorry, I'm not sure I understand.
You said it is underestimated that prediction. I mean, observed data is underestimated then prediction, right? Observed chlorophyll
with the neural network?
Yeah,
it tends to underestimate the signal, yes, because the difference between observation and prediction is in blue.
What percentage?
Percentage?
Yeah.
I'm still working on this neural network because this project was part of a NASA project, and I want to maybe publish those results. And so now I'm working on comparison with another model to be able to say if those predictions are enough good or no. So I'm still working on that. I don't have the percentage of difference, but okay. Maybe the intensity of the signal is not the most important. What we really wanted to know if where the peak of fluorescence is located along the water column. So, but this result seems to be good when we are comparing observation and prediction.
Yeah. Have you tried this predicted chlorophyll with satellite data in surface?
I'm working on that.
Okay. Okay. Thank you. Thank you. Any other questions?
I have a
Can
small
I
question.
ask one question?
Yeah. Please.
Shall I ask?
Lucia, it is a very nice presentation.
Okay.
So I just wanted to know that whether this predicted chlorophyll errors is due to the non-parameterization of other factors like light or traits.
I don't have a good connection, sorry. You are suggesting to test non-parameterized analysis, is that?
Yes, yes, because light and nutrients also a factor for this chlorophyll variability in this polymer region. So this, so my question is, if that is the case, whether we can use only this temperature and salinity for the prediction of chlorophyll in this polymer region.
Yeah, sorry. Yeah, I didn't understand all the question. Like,
Okay. Can you, Dr. Sabu, can you just chat with her?
Yes.
yeah.
Yeah. Okay. So any other question, or we'll move?
I have a small question.
Yeah, please.
Actually, any water masses has been identified in this area?
What?
Water masses.
Marta?
Water masses. Water mass.
Water masses. I want for another polynia, Cardane polynia, we really want to add water masses to try to explain more what we saw for this polynia. It's also why I didn't present the results for this polynia, because I think we still have to add water masses to our results to be able to interpret. And so when I will do that, I will also add the water masses for the Shackleton polynia. And yes, so this project, I did this project for my master's degree, so it was just a 5 months internship, so it was quick. And I'm still working on that, and of course I don't have all the results now. So yeah, but I'm thinking about the water masses. Yes.
Thank you.
Okay. Thanks, Lucy. Then we can move to the next speaker. Next speaker is Rafael Henrique Moro-Falcio. And the title is Vortices Effect in Phytoplankton Community at Brazil, Malvinas Confluence.
Hi, hello, can you hear me?
Yeah.
Okay, so I'm not exactly Rafael, he had a problem, so I'm one of the authors, I'm substituting him in the presentation. I will share my screen. Can you see the presentation?
Yes, we can see.
Thank you. So, let me go back here. Okay, so hello everyone, my name is actually Gabriel, and I will present this in behalf of Rafael Falcão. And this presentation named the Vortex Effect in Phytoplankton Community at the Brazil Malvinas Confluence. This presentation is part of the Mephisto project, Antarctic project from Brazil. And this was a working collaboration with me and Rafael Falcão and the professors Maria da Glória Cunha, Moacir Araújo, the bachelor Pedro Reis, and the professor Pedro Melo. So, talking a little bit about the Brazil Malvinas Confluence, is the area of encounter of these two really contrasting different currents. The Brazilian currents that run from north to south, that has a higher temperature, higher salinity, and lower nutrient concentrations, and the Malvinas current that came from south to north, and has a very lower temperature, lower salinity, and increasing nutrient concentration. So these two currents that also carry different phytoplankton communities encounters here near to the Rio de la Plata plume. So all the encounter of the plume in these two currents create a really high energetically active area. That's also a hotspot of diversity and phytoplankton biomass, and as a really energetic area, formate several eddies here from both warm and cold eddies with water of these two currents. So these eddies, as I say, it can be a cold core eddy if the water trapped inside is mostly composed of the Malvinas current, or a warm core eddy if this water are from the Brazilian current. So these two types of eddies have different nutrient concentrations that will create different productivity and different community structures of phytoplankton. So the object of this work was to assess this community composition of the phytoplankton in these two different eddies, a cold core eddy and a warm core eddy, and in the Brazil-Malvinas confluence, testing the hypothesis that this cold and warm core eddy change both phytoplankton abundance and composition in comparison with the Brazil-Malvinas confluence and these original currents. So we realized an oceanic campaign in 2019 in Auschwitz Spring, sampling in 11 stations, 4 of them inside the area of the Brazil-Malvinas confluence, with 2 really inside the Malvinas currents and 2 inside the Brazilian currents, and other 7 stations here inside different vortices, with 3 inside a warm eddy and 4 inside a cold core eddy. So the phytoplankton for both qualitative and quantitative estimations were realized with Niskin bottles in a rosette in 2 different depths, in the surface and on the deep chlorophyll maximum. And we also had information about temperature, saline conductivity, and nutrient concentrations that could not be used in analysis here because the information came just a little bit later. So in results, in the physical chemical information of the ore masses, the Brazilian current had higher temperature, salinity, and dissolved oxygen than the Malvinas current, with an average of 15 from the Brazilian current, and here 6 in the Malvinas, and also here a higher dissolved oxygen concentration. And so on, the eddies had similar characteristics to, could say, its father currents, with the warm core eddy having temperature, saline, and oxygen concentrations similar to the Brazilian current, and the cold core eddy having the same parameters similar to the Malvinas current. On some phytoplankton results, we observed 62 species in both the confluence and the vortexes, with the diatoms Bacillariophyta being the most diverse group, with 32 of the 62 species, followed by dinoflagellates with 25, and then Ochrophyta, Ptophyta, and Cyanophyta had lower diversity. In abundance, we could observe that in both the surface and the deep chlorophyll maximum, the Malvinas current had higher abundance than the Brazilian current. Here you can see the surface, and here in the deep chlorophyll maximum. But the higher abundance we could observe was inside the cold core eddy, that had an abundance of 93 plus 10 at 8, and the lower in the warm core eddy. So this could be expected since the eddies can trap cells inside them. This could be the case here of the cold core eddy, although here in the warm core eddy, this really lower abundance could be an answer for the lower nutrient concentrations that could happen in these warm core eddies, although in this campaign, only inside the Malvinas current, we had a significantly higher nutrient concentrations of nitrate and phosphate. So here, if you look at the community composition, this can clearly be reflected, since the Malvinas current was the only one that had a contrasting community composition than the other regions. The Malvinas current were mainly dominated by diatoms, especially minidiscus and pseudonychia pungens, and both current of Brazil, the cold core area and the warm core area were dominated by dinoflagellates, especially Calodinium. So we did an interval to see if some of the species that we observed could serve as an indicative for the depth, the surface, or the deep chlorophyll maximum, or the different currents or eddies. So these are results. In blue are diatoms, so mostly diatoms can be used as a indicative for the different water masses. And here we can see that the area that had more species that could be used as indicative is the Malvina current that also had the higher diversity. So between eddies to the warm core eddy, only Planktonella sol can be used. It's a small diatoms typical from tropical waters, and from the cold core eddy, especially Plaginolepis carginesis, which is a temperate species, and this also could indicate this difference in communities trapped between these two eddies. And we also try to see if there is how this community will change between the Brazil Malvinas currents, this first column, and the two eddies. So this is the most important, most abundant species in the Brazil Malvina currents, and how they change between the warm and cold core eddy. So we could see that in the warm core eddy, most of the species had a reduction in its abundance. So here in red, with only Asadino and Dicnota fibula having an increase in abundance from the Brazil Malvinas current, Malvinas confluence, sorry. And the cold core eddy had increase of more species, with the increase of some large diatoms and some colonial species. So this is a dendrogram of our samples, and here we can see that they are well grouped. So here we have the samples from the warm core eddy, here's from the cold, and here's is from the Brazilian current, and here from the Malvinas current. And although in the cold core eddy we had a slightly higher nutrient concentrations, although not significant, and the presence of some species characteristic of temperate and some polar water masses, we, and seen by this dendrogram, imagine that this vortex is actually more associated with the Brazilian current, which could also be seen by the community composition in general more similar with these currents. So although we can't still say what is controlling what current is more related with the formation of these two vortices, we will have more campaigns this year. So we hope that with more information, more stations, and more data, both physical, chemical, and also nutrients, you can have a better look of the effect of these eddies on the phytoplankton community of the Brazil Malvinas confluence. So here, in conclusion, we observed that the Malvinas current had higher nutrient concentration than our other areas, and with this helped with the domination of the diatoms and higher diversity and abundance. And both areas had a different community composition from the region of the confluence, and we imagine that the Brazilian current had a higher impact in the formation and the composition of these areas than the Malvinas currents, although this you need more information to say it with sure. So I want to thank the organization of the SCAR, the Federal University of Pernambuco, CAPES and CNPq for definition of these campaigns, and especially the Mephisto project. If you want to see see a little bit more of the all other groups that are working in this project, you can search for Mephisto or Prontar Nordeste in Instagram or social media, YouTube. You have a lot of YouTube videos, so feel free to know more. Thank you.
Okay, thanks. Thanks, Rafal, for your nice presentations for this phytoplankton various community in Brazil Malvanis confluence. And any questions? Any questions from audience or panelist?
I have one question.
Yeah, please.
Yeah, you have told here two currents, that is Brazil current and
Yes.
Malvinas current.
Yes.
So Brazil current with the warm core eddy and Malvinas current with cold core eddy, right?
Sorry, I didn't understand.
Brazil current, you are
Yes.
observing the warm core eddy, WCE.
Yes, the Brazilian current is a tropical, warm current from the north, and the Malvinas current is, and the sample was in the region of the confluence of these two currents that form these vortexes close to Argentina coast.
So that is the reason for this warm core and cold core.
Sorry,
Warm core eddy you are getting with the Brazil current.
Yes.
And cold core with the Malvinas current.
Yes.
So because of these two currents are, one is warm current, one is cold current.
Sorry, I can.
No, what is the reason for these different eddies?
What is the
reason for formation of this different eddies in these two currents?
What is the reason for the formation? The reason for the formation.
The reason for the formation of
okay. Yeah. Great. No, it's the impact of the two currents go in direction of each other, and this create these mesoscale structures that go in direction from the middle of the Atlantic. So of course, it depends in how these mesoscales structures form. You can have the trapping of the Brazilian current waters. So this will be a warm core eddy, or the trapping of the Malvinas current. This could be a cold core eddy. So in this study, by the physics of the eddies, the temperature, we think that one was a warm and one was a cold core eddy. But looking at the phytoplankton community, actually both of them looks like communities of a warm core eddy. So this is why we maybe need to look a bit more of how these are formations. So maybe not only temperature can say if this will be a warm or cold. In the future campaigns we can learn more.
Okay, thank you.
Rafael, I have one question.
Yes, please.
That's this cyanophyta is widely distributed in global.
Yes.
But the cyanophyta population is very less in this Malvanis confluence, what you identified, right? So what is the reason? What is the reason of this less population of
lower importance? Yes. So really, if you look inside more, in higher latitudes, there's really a lot of cyanophysi. But I think that here, as we have this higher productivity, in these cases, normally like diatoms and dinoflagellates have a increase in abundance. And maybe if you look at the pico and nano phytoplankton, you could see these higher concentrations of cyanophysi. But here we only have data from the microphytoplankton. So could be this. We're just not looking at the right zone to see the cyanophysi. So this is just a cut of the microphytoplankton.
And another question, can this diatom in warm core eddy can sustain in a cold core eddy, the diatom species, what you find?
Can sustain what?
Sustain in cold core eddy or not?
Yes, so the diatoms are mostly in the cold core eddy. So yeah, they are dominating community there, and I don't know why we don't see them in the warm core eddy, but this is something that we need to study a bit more. Okay, thank you.
Thank you. Thank you. Any more questions? Okay. Then we can move to the next speaker, Catherine Becker. And the title is the role of diabetical migration of gelatinous zooplankton in downward carbon export, a Southern Ocean mesopelagic migrant form case study. Catherine is there.
Sir, we have a pre-recorded talk, we can play that.
Okay, so now we can, two records are there. I think one is Nicole and other is Catherine, right?
Yes, sir.
So first we can play the Nicole and then Catherine.
Yes, sir, we can do that.
Okay, please start.
Hello everyone, my name's Nicole and today I'll be talking to you about krill swimming behaviour in response to chemical, photic and physical stimuli. So why do we study krill? Well, we know that krill are a key species in the Antarctic ecosystem, forming that major link between lower and higher trophic levels. We also know that krill form large schools and swarms, but the behaviour of why they form those swarms, why they dissipate from those swarms, and the structure of those swarms themselves and the behaviour within those swarms is all largely unknown. We have a lot of acoustic data, both from the fishery and from scientific voyages in the Southern Ocean about krill swarms. So we know the size, the shape, the density, and even the depth that we regularly find them at. But we don't know, again, a lot of the environmental conditions that might make a swarm form. We also know that krill are very vulnerable to climate and environmental change, and this might impact whether they're even able to form swarms, where they can form swarms, and whether predators or even their prey will be impacted into the future. So to be able to predict this better, we need more data to help improve our models. To do this, we're actually using video analysis of three-dimensional tracks of krill in a horizontal flume. So a horizontal flume is just a sideways cylinder of water with an inlet valve at one end in the horizontal direction. This inlet valve allows us to control the physical stimuli or the flow rate of the water, as well as our chemical stimuli. So we have a negative attractant, which is guano or penguin poo, which is our proxy for having a predator nearby, and chlorophyll, which is a positive attractant, which is a proxy for having some food nearby. We also have our photic stimulus, which is a light, which we can set to both bright, which is a surface level light, and dim or dark, which is at 100 meters depth. We then have two cameras set up in stereo, one at the top of the tank facing downwards and one at the side of the tank facing through the flume. So our top camera records the X and the Y coordinates in the tank, and our side camera records the Y and the Z coordinates in the tank. And by matching up the frames of those two videos together, we can then get a three-dimensional track for each frame of those videos. Some of the questions from that project are, what are the relationships between krill movement speed and the direction to flow? And can krill do things such as perform area restricted searches to be able to find their food? Does this change depending upon if there's flow, chemical, or a combination of the two? Can krill navigate in the dark? And can krill avoid predators? And can they avoid predators in flow or in the dark? So this is just a small number of the questions that we're hoping to answer in this project. So looking at some results, you can see that the turn angles of the krill within the flume vary wildly depending upon what variable there is. Now, this was also a multifactorial project, so we actually have some of these factors at other levels of other factors. So let's delve into this in a bit more detail. So you can see on the right here, we have flow at 0 centimeters a second and flow at 0.6 centimeters a second. And we tested different chlorophyll levels at each of these levels of flow. And you can see that as chlorophyll increases, we get not only more orthogonal turns, so that's the left and right turns at 90 degrees, but we also get a greater reduction of those smaller turn angles. So krill are in general doing much larger turns. Whereas in those lower chlorophyll levels, we're actually getting krill swimming with much smaller turns and less orthogonal turns. And this might be because with less chlorophyll available, they need to do smaller turns to be able to stay on the krill patch. Whereas krill at the higher chlorophyll can do much more orthogonal turns to be able to find the densest area of that chlorophyll. However, at a flow of 0.6 centimeters a second, you can see that the highest chlorophyll levels actually have the smaller turn angles, whereas the smaller chlorophyll levels have lower turn angles, which is the inverse of what we see at flow equals 0. We also see that there are more orthogonal turns in the higher turn angles. So potentially this is a case of krill finding where the krill patch is and then trying to maintain it, particularly in those high chlorophyll levels when there's a flow available. Potentially the inlet valve giving them a directional way to find where the chlorophyll is meaning that they are then doing smaller turns to maintain that heading. And there's a really good way of being able to tell that, and that's by looking at their headings. So obviously the turn angle and the headings are very closely related to each other. So if you do a large turn, you're going to change your heading a lot greater than if you do a small turn. So we can see again that depending upon what the variable is, that there is a lot of difference here in the headings that the krill are swimming in. But again, we can break this down further. Again, looking at flow of 0 and flow of 0.6, we can see that as chlorophyll increases, the krill are more likely to be swimming in those orthogonal directions of left and right in the flume than they are up and downstream. In fact, krill are more likely to swim both up and downstream in a flow of 0, so they have no directional bearing from flow, even with no chlorophyll present. This may be because there was still a passive amount of water going through the flume, even though there was no active pumping of water through those channels of the inlet valve. However, interestingly, at a flow of 0.6, we see that as the chlorophyll increases, that directional heading gets stronger and stronger towards that inlet valve, as well as the downstream direction. We see less orthogonal turning even in a chlorophyll of 0, and we see more orthogonal turning in those higher chlorophylls. So we're already starting to see some of this coming through. Again, we can also look at krill velocity. So this is all the velocities in relation to the flow. So you would imagine that a krill swimming in a flow of 8.9 centimeters a second in one direction would have to swim at 8.9 centimeters a second in the other direction to be able to maintain their position within the video itself. So even though our initial velocity that we get from our analysis is 0 because they're not moving in the frames of the video, once we correct for that flow rate, you can see that they are roughly matching the flow of the water. So again, we can break this down further. So at a flow of 0, we actually see that as chlorophyll increases, the krill are actually staying where they are. They are quite happy. They have lots of chlorophyll right where they are. There's no current to push them anywhere. There's nothing pushing the chlorophyll away from them. So they're just going to stay where they are and maintain a velocity of close to 0. However, in those mid levels of chlorophyll, potentially the krill are trying to find a better patch of chlorophyll. Potentially they're trying to find more prey. But it might also be a case of the concentration isn't quite ideal for them to feed on, so they have to swim around a bit more to be able to actually gather up that chlorophyll that is available to them. So potentially this is them trying to search to find the better prey field. So we see them increasing their velocities in those middle chlorophyll levels. However, as soon as you introduce flow, we see that not only are they matching the flow of the water at 0.6, but regardless of what the chlorophyll level, they're all using that same swimming velocity, that same swimming behavior of trying to match the flow. And we already know that they're trying to head in that same direction, flow of 0.6 as well. So the behaviors between different chlorophyll levels at that flow isn't very different. Getting into the modeling. So taking the log of the velocity, we can actually start to look at how this affects our stepwise modeling program. So we have the log velocity of time 0 on the x-axis, and then the log velocity at the time of the next step in the model. And in this case, it's the next frame of the video for our data. And you can see that there is a strong relationship there between the two. So the velocity that krill were going in one frame highly correlates with the next frame. Using this regressional coefficient, we can actually start to look at how that differs for different factor levels. So we can see that as chlorophyll increases, that coefficient goes down. And what this means is that there is more swimming variability in terms of their swimming speed in higher chlorophyll than there is in lower chlorophyll. So krill in 0 chlorophyll will quite often just swim around at the same speed. They're not doing anything. There's nothing impacting them. So why would they need to change their behavior? Why would they change their swimming speed? Whereas krill in those highest chlorophyll levels are immediately changing their swimming speed far more often. And this might be because of something like an area restricted search where they're swimming quite quickly. trying to gather up the chlorophyll when it is present, swimming quite slowly when it's not present, or potentially swimming fast to be able to get to the ideal patch and then swimming slowly once they're in that patch. But there's more variability there than what we see in zero. Again, with the flow rate of krill, we can see that as the flow rate increases, the variability of the krill swimming speed goes down, so the coefficient goes up. So to be able to match that flow rate, obviously they have to swim at a particular speed pretty consistently to be able to match that flow, which means that there's not much variability within that velocity of the krill swimming, which means that they have a very high coefficient. Whereas at a flow rate of zero, where the krill have free rein to be able to swim wherever they want, they have a lot more variability. We also sent out a survey to some key stakeholders to find out what sort of things they would like to see in the model, where they see it going, as well as if they would be interested in something like an interactive web tool. So some of the questions included, would this be useful for your current or future work? We were very happy when we found out that 7 out of 8 respondents actually said yes. We also asked what kind of input people would like to be able to put in, whether they would want to be able to put in their own data or just be able to play with some of the variables that we already have in there. What environmental variables were most important to them in their work? So obviously we haven't tested for every kind of environmental variable. We've only done a select few, but we're always hoping to expand that to be able to do more experiments with more conditions. What model outputs would be useful for you? So again, a lot of the outputs that we're getting at the moment are at a very individual krill level, but we would love to be able to include more to do with the aggregation and the swarm structure itself. This is a very simple model. It's using NetLogo. This is mostly just to visualize what the model is like. The model itself is very math-based. So this helps visualize it, particularly for me. I know I'm a far more visual person. But you can see that depending upon the velocity of the krill, how far they can see, so that vision, whether they're more spaced out, whether they want to be more in the center of the world or more towards the edges of the world, they have a more natural flow and rhythm to how they're swimming. So where to from here? Well, this is only data from our horizontal flume. We also have a vertical flume, so we can test for upwelling and downwelling and how that impacts on the krill swimming behavior that we're actually taking to Palmer Station this season. So we're very, very interested in seeing how that affects the swimming behavior of the krill. But we're also very interested because there is a side port in the vertical flume, which is actually how we get the krill in and out of the flume, that they could potentially use as a refuge, not only to get away from things such as guano, as a refuge away from predators, but potentially also to get out of high flow conditions, even if that flow is in an upwards or downwards direction. So we're very interested in seeing how that impacts on their swimming behavior. Ideally, we want to get to a point where this model can actually be 20 to 50,000 individuals, and that we can then overlay it over a large oceanographic model. Obviously, we're still a while away from that, but stay tuned. This is also data just from individuals, as I said before. So potentially there's things, behaviors that we're missing that you would be able to get from collective krill tracks and collective krill data. And there is a concurrent project being run at the University of South Florida, and Kuvat Gariev is actually presenting a poster exactly on that topic and the research that they've done. So anyone who is interested in that side of the project, I would highly recommend going and seeing Kuvat's talk and poster. Thank you so much for listening. If you have any questions either about the experiments themselves, what we're doing at Palmer Station this season, or how the modelling is being developed, please feel free to contact me either via email or Twitter. And lastly, just a big shout out to everyone at Palmer Station, the RV Guild, and all of our undergraduate students who helped us wade through the monumental amount of video analysis that had to be done.
Thanks, Nicole, for your nice video presentation. So if any questions, we'll contact you, as you say, through your email. Okay, e-poster presentation, quick presentation, that is 3 minutes. So I'm seeing in this chart, there is 2 people are there. I think the first is Anna Cecilia. So can I ask to Anna Cecilia, will you present your posters? Anna Cecilia is there?
Are you listening to me now?
Yeah.
Okay.
Do you want to
I'm
show the?
having some connection problems here at my home, so I ask you sorry.
Oh, it's okay. So now you want to show the presentation? Yeah.
Can I share my screen?
Yeah, please.
Okay. Are you seeing the screen?
Yeah, it's visible.
Okay.
Yeah.
I'm having some problems here with the connection. Sorry, it's my
Oh, it's
computer,
okay.
I think.
It's okay.
So first of all, I will thank you for the opportunity of presenting my work at SCAR as a post farm. I'm Ana Cecilia. I'm a professor at Federal University of Bahia in Brazil. And here I'm gonna presenting the first data we have, the preliminary data we have from our work that it's in the same project that Rafael just presented 2 presentations ago. So they did they presented the data that we had in the Malvinas and Brazil currents confluence, and we also have some samples from the Bransfield Strait in Antarctica. That it's what I'm going to present here. My work has as main objective evaluated the organic matter composition and the changes of this organic matter between the Bransfield Strait and the Admiral Bay that is in the King George Islands in Antarctic Peninsula. For this, we made some suspended particulate material samples. For we sampled surface water and water in the maximum chlorophyll depth, and we filtered this water. And in this suspended particulate material that we made some evaluations of the total particulate or organic carbon values, the total nitrogen, organic nitrogen particulate material, and the isotopic composition of the nitrogen and the organic carbon. We made this in a elemental analyzer coupled to a isotopic ratio mass spectrometer. The samples were done in 2 distinct days. In the first day, we made the samples just in the beginning of the inside of the bay here. And in the second day, we made in front of the bay in the Bransfield Strait. And what we saw, our results showed that the particulate, this is a very preliminary data. And for now, what we can see is that the organic matter in the surface sample and in the maximum chlorophyll depths are the same. There were no difference between the particulate organic carbon content, the particulate nitrogen content, and the isotopic composition. It was the same. However, there was some difference between the data that we found in particular for the CN ratio and for the nitrogen content in the samples made just in the beginning of inside the bay, just here, and in the Bransfield Strait, showing that in Bransfield Strait we have a relative contribution of phytoplankton in this organic matter higher than in the samples that was in direction of the bay. And what we think that can making this is the mosses contribution. So the main source of organic matter for all samples are phytoplankton, but we think that the mosses are contribution for this difference that we observed in the Bransfield Strait and in the Admiral Bay, especially because of the carbon isotope composition that were a little bit more depleted than the values that are commonly found in the phytoplankton. We are gonna do this year another set of samples we are going to Antarctica doing another samples to complete this analysis. And we are also going to do some organic markers as alkanes, sterols, fatty acids that will help us to verify this hypothesis that we have, that the monsters are contribution a little bit with the organic matter that we can find in the Bransfield Strait and the Admiral Bay. So I would like to thank you from our project, Mephisto, Plantar Nordeste. Here I have I put some social medias that we have. This is a part of a big project, and if you want to see more and know more about this project, we have the website that is here. We have a YouTube profile and Instagram profile. I would like to thank you also for the 4 Women in Science program from L'Oréal and UNESCO that is providing some sources for we doing this project. CNPq and Atlanteco that is also supporting this project. Thank you. Are you listening?
Mishra? Mishra, can you hear?
I'm here. I'm listening.
Okay. So you are telling that the phytoplankton is the main source for this particulate organic matter, right?
Yes, it is what is commonly found in the area. There is another works done in other bays from the King George Islands, from a research group from Uruguay that has already found that phytoplankton is the main source of organic matter to the area.
That means
however, the isotopic composition that we found was a little bit different, and the CN ratio shows that something else is happening.
So this is diatoms?
I don't know yet. We are going to make sure of that with the organic matter, organic markers that we are going to evaluate, the sterols and alkanes. This is a next step, conclusion that we can make before this analysis. This is very preliminary analysis, and we are very excited because it was different than we were expecting. So maybe in the next SCAR, I can show everything. And make sure that
thank you for the
phytoplankton contribution and mox's contribution.
Thank you.
Okay.
I have one question. What is your depth of a water column?
The depth of the water column, it's changed a lot. It goes from 100 meters to 1,000 meters.
No, but what your sample is collected from up to which depth?
In the water column, it changed too, because the maximum depth chlorophyll, it changed a lot during the sampling, but it goes to 20 meters, 50 meters, something like that. So I don't remember by heart now, but it's something like that.
You got the homogeneous, the POM you got homogeneous?
Yes,
in the entire column? Why? What is the reason? Anything? Any reason you can explain? Homogeneous.
I'm so sorry. There's some noise here with my neighbor. It's the computer that's not working, the neighbor that is not helping. Could you repeat so?
Okay. I want to ask, POM were homogeneous in the water column, right?
Yes,
POM.
our data show us that the water column is a little bit homogeneous. We also have some data of nutrients there that show the same.
Any reason why it is homogeneous? Why it is homogeneous?
I don't know yet. I don't know yet.
Okay, we'll discuss later on.
Okay, maybe in the next car it will be more complete. This works.
Okay. Even we can discuss by your mail or anything else if we have. Okay. So can we go for the next poster presentation, that is Cuvette Garoff. Cuvette Garoff is there.
Hello, I'm here.
Okay, please.
Hi.
Can you close, Anna, your this? Yeah.
I
Yes,
cannot
yes.
start it unless another is closed.
Your title is a novel annular flume to measure schooling behavior of Antarctic krill, right?
Yes. Can we close the other one?
Yeah. Can I ask IT team, please do change?
No. She has to. Anna has to.
Okay.
Close it.
Anna, would you please close your sharing?
Okay. Should be good.
Yeah. Now you can give it. Yeah.
Yes. So in 3 minutes, I'll
Please.
try to give
Please
high
go ahead.
level overview of my work. So
Yeah, it's
basically,
visible.
it's a methods poster about novel annular flume to measure schooling behavior of Antarctic krill under various environmental cues. So Nicole presentation talked about individual Antarctic krill behavior under different environmental cues such as chlorophyll levels, light levels, flow rates, etc. Now, in our case, we'd like to understand how Antarctic krill as a school respond to this kind of environmentally relevant cues. So for that, we have built a laboratory annular flume. So there are a lot of parts associated with this setup, but I will kind of give high-level explanation of each part. So this is our flume, annular flume. So the reason we have annular because in a rectangular flume, the krill might be stuck at the upstream or downstream at those grids. So this annular flume allows for continuous schooling without encountering any wall. So there are mainly 3 parts for this flume, basically flow creation. So the flow is created with the drum rotation at the center. So if I come here, so the shaft rotates the drum at the center, and that creates a the flow at the inner radius. And then there are up to four submersible flow conditioners. These are 3D printed channels that can create tangential jets at the outer radius, at the outer wall. So with these 2 boundary conditions, we can create some annular flow inside our tank. So that's how flow is created. And then now we have to also characterize the flow, meaning we have to know at what pump flows, at pump conditions and drum rotations, what kind of flow speeds we are looking at. So to do that, we used particle imaging velocimetry. So the idea is to send laser sheet at our tank at different heights, and then by measuring the speed of those particles inside the water, we can get the profile, speed profile in our annular tank. So for example, at high flow, which is shown in green here, so we have around 40 millimeter per second speeds. So this graph shows the profiles in our speed profiles in our tank. So this gray shows inner quarter of our inner drum, and then this another gray arc shows the outer wall. And also we have low flow at around 6 millimeter per second, as you can see. So this is how we vary the flow rate and how we measure the flows in our tank. Okay, then the next step, the big idea here is to measure 3D tracks in our tank. So for that, we use 4 cameras. So one camera looks at entire tank and 3 others look at the shared field of view. So in blue here, you can see all these 3 cameras look at the same field of view. And then by using calibrations, we can get 3D trajectories of our krill inside our tank. So for calibration, we use some methods available called sparse bundle adjustment. And then for detection, so detection is quite hard because there are hundreds of animals in our tank, and it's really hard to do this manually for 5 minutes of recording. So we use deep learning software called Loopy. So it's hard to see, but we can get head points of each individual krill and then tail points. So once we have all the detections in camera coordinates, we can use calibration coefficients to get our 3D tracks. So these blue lines show blue trajectories show the krill motion. And then from here, we can get school structure parameters such as speed, nearest neighbor distance, and polarity, etc. So after that, so we sent this setup to Antarctica end of 2019. And we got some data, and here are our results. So at 3 different flow levels, one of them is no flow, others are low and high flow. So these are 12 millimeters per second, and this is around 30 millimeters per second. And we plot swimming speed as a function of different flow levels and then light levels. So we have around 30 millimeters per second as a swimming speed, which is around 1 body length per second. And then we can plot nearest neighbor distance. That's also around 35 millimeters per second. That's 1 body length per second. So you can see that the relationship between all Qs, flow levels, and light levels are complex. And then we can plot other relevant parameters such as polarity, for example, how aligned these individuals are. So as the flow rate level increases, we see that polarity increases, which means they are more aligned with the flow. And then NGDR also increases, which shows the tortuosity of krill paths. So in general, we see that relative speed, which is the speed of krill relative to water, increases with the flow, but not proportionally. So which means as we increase the flow rate, they cannot resist the flow anymore. They cannot swim faster to keep up with the flow. So it's not proportional with the flow rate. And then nearest neighbor distances didn't change much with light or flow levels. And flow result in polarization of krill with more straight paths. That being said, I think I forgot something here to talk about, which is how we create a light. Okay, so Nicole also mentioned that we want to see how krill schools change at dark levels, dark light levels, which is 100 meters below the ocean surface. So in those levels, we cannot see krill because it is completely dark. So for that reason, we put backlighting, which consists of near infrared light strips attached to the underneath the tank. So since krill are not sensitive to this near infrared light, but cameras are sensitive, so we can still see krill even in the dark. So I think that concludes, and we will shipped this setup. We already shipped this setup to Palmer Station, Antarctica, and we will be doing another set of experiments this fall to get more idea about how guano or chlorophyll levels affect their school structure parameters. So that's all. Thank you for your attention. Yeah, if you have any questions, you can ask.
Mishra.
Yeah, thanks. You have any questions? Anybody have any questions?
Yeah, I have one question.
Please.
Actually, you are telling that the krill is not not proportional with the flow rate, right?
Yes.
So in the ocean, there is we can expect currents.
Yes.
So it is not proportional with that. It is not flowing in that direction.
Yes, I think what happens is they can swim so much, so they cannot, for example, swim too fast. They can swim up to some point, and then basically they will be drifted with the flow. But they have some capability to swim against the flow, but not too much. So that was the idea of that plot.
Okay. Up to what depth we can expect krill?
I think one paper specifies, one paper has a plot that has swimming speed as a function of flow. So they can swim up to, I believe, 8 body lengths, maybe even more. So that would be like 300 millimeter per second.
Okay. What is the depth we are expecting that krill existence?
Depth?
Krill, up to what depth? In the ocean?
I think
100 meters or 200 meters?
I believe mostly they can, I'm not sure, but I know that they can school maybe around 200 meter,
Okay,
but maybe they can go deeper. I'm not sure.
Okay, thanks. Thanks for your this finding novel annular film to measure schooling behavior quantitatively. And anybody have any further questions can contact to Cuvette care of. So with this, I'm not seeing any other people for the poster presentation. So we can conclude here. So I can ask to Christina Weldrick, do you have any comments or suggestions? Christina.
Actually, I think Edward is here. Edward Menschel.
Oh, do you have any further comments, suggestions?
Oh, no, sorry. I was just commenting on the fact that I see another attendee here that is part of the poster presentation lineup.
Okay.
But no, as far as the talks, everything has been really wonderful. Thank you for the talks.
So thanks to panelists and thanks to speakers, all speakers, audience. Since last 2 days, we have this session discussion. This is our session, Southern Ocean Plankton Diversity, Food Web Ecosystem and Biogeochemical Cycles. So it was really, all presentation was very interesting and it was very useful for this Southern Ocean ecosystem and biogeochemical cycles, biogeochemical processes, even linked to climate change. So I can finally, thanks to this SCAR Open Science Conference and NCPOR, Ministry of Earth Sciences, who provided this platform to discuss us with Neslan international participants for understanding our Southern Ocean plankton diversity and food web ecosystem in Southern Ocean. Probably in near future, if we may have opportunity to discuss again with our such type of research and finding on this Southern Ocean. So it was a really is opportunity to progress our this Southern Ocean ecosystem and biogeochemical study of Southern Ocean ecosystem and biogeochemical cycle. So can I ask anybody else have any comments or anything else? Yeah.
Yeah, Mishra, actually, all the presentations and informations what we gathered from here is wonderful. And when we are seeing that other regions, we are doing in the Indian sector of Southern Ocean, but other regions also good studies are being carried out.
Yeah.
And we got the information what they are doing there. And this is an inspiration for us to conduct the more understanding about the Southern Ocean sector. So this is a very good experience for all of us. Thank you for all speakers and the keynote speakers. Thank you so much.
Okay. Finally, thanks to this IT team who all cooperated us to conduct this conference successfully. Okay, then can I ask Dr. Sabu? Will have you can take any snapshot for us?
Yes.
So I would like to request all please switch on their video.
Please switch on your video.
Yeah,
so
please switch on your video. All panelists, participants, yes. So we'll have one snapshot.
Yes. Martina video is not on.
Yeah, Martina Messioni. She's there. Yeah, she's there. No. Okay. Any other?
Just a minute. Yeah, I'm taking. Yes, done.
Oh.
Thank you.
Thank you. Thank you. See you.
See you all.
See you again in future.
Thank you,
Okay.
Christina, and all others. Thank you so much.
Thank you.
Thank you.
Thank you.